In this video session, we'll be talking about organogenesis. Organogenesis is the phase of embryonic development that starts at the end of gastrulation and continues until birth. During organogenesis, the three primary germ layers which are formed from gastrulation, that is ectoderm, endoderm and mesoderm, forms the internal organs of the organism. The cells of each of the three germ layers undergo differentiation, where less specialized cells becomes more specialized through the expression of specific set of genes. And cell differentiation is driven by cell signaling cascades. Differentiation is influenced by extracellular signals such as growth factors that are exchanged to adjacent cells, which is called juxtacrine signaling, or to neighboring cells over short distance, which is called paracrine signaling. Intracellular signals consist of cell signaling itself, which is known as autocrine signaling. They also play a role in organ formation. So the main role of signaling pathway is to allow for cell rearrangement. It also ensures that organs form at specific sites within the organism. The organogenesis process can be studied by using embryos and organoids. Now let us first discuss about the organs produced by the three germ layers. Endoderm is the innermost germ layer of the embryo, which gives rise to gastrointestinal and respiratory organs by forming epithelial linings and organs such as liver, lungs, and pancreas. The mesoderm or middle germ layer of the embryo will form the blood, heart, kidney, muscles, and connective tissues. The ectoderm or outermost germ layer of the developing embryo forms epidermis, the brain, and the nervous system. Now let us discuss about the mechanism of organ formation. Each germ layers forms specific organs. In the 1820s, embryologist Hans Christian Bender discovered that the germ layers cannot form their respective organs without the cellular interaction from other tissues. Internal organs began to develop within 3 to 8 weeks after fertilization. The germ layers forms organs by three processes: folds, splits, and condensation. If we have to talk about individually, then folds are formed in the germinal sheath of the cells and usually forms an enclosed tube which you can see in the neural tube of vertebrates. And splits or pockets may form in the germinal sheath of the cells, forming vesicles or elongations. The lungs and glands of the organism may develop this way. A primary step in the organogenesis of chordates is the development of the notochord, which includes the formation of neural plate and ultimately the neural tube in vertebrate development. Development of the neural tube will give rise to the brain and spinal cord. Vertebrates develop a neural crest that differentiate into many structures including bones, muscles, and components of the central nervous system. Differentiation of the ectoderm into the neural crest, neural tube, and surface ectoderm is sometimes referred to as neurulation, and the embryo in this phase is the neurula. The coelom of the body forms from the split of the mesoderm along the somatostasis. During organogenesis, the main changes that occurs in the vertebrates are elongation of body, formation of tail, division of the body into head and trunk, separation of the embryo from the extra-embryonic parts, that is formation of different organs from primary germ layers through the process of differentiation.